So we're going to be talking about biomolecules, or otherwise known as macromolecules. So these are the things that make up living things, okay? And really the crux of macromolecules comes down to carbon, okay? So these are all organic molecules, which means for our purposes that it contains carbon. Now, remember that carbon is super versatile. It can bond with itself, it can bond with hydrogen, it can bond with just about anything, and form these really, really complex structures. So here are some examples down here. So here we have a triple bond between carbons, and then some hydrogens attached on either side, some double bonds, some single bonds, etc. Okay, so there's some real diversity here, and this is really just the tip of the iceberg. Now, when you start talking about organic compounds inside of living organisms, it's really best to think about it in terms of 4 categories. Okay, they do interact sometimes, but there are 4 categories. Okay, so we have the amino acids, and those amino acids are one piece that makes up the puzzle that is proteins. Okay, so we're going to string together some amino acids to make a protein, and I want you to just kind of think about that, because that's going to be a trend that we see again and again. Things being linked together in order to form bigger things. And then we also have fatty acids, and when you take fatty acids, you can combine them to make other kinds of lipids. Okay, so like phospholipids. We're going to talk about different examples. We have carbohydrates. Now, carbohydrates are a little interesting because they have subunits, and then those subunits can be connected together into what we call polysaccharides. But we also have some that just kind of hang out by themselves. Okay? And then here we have nucleobases, okay, or nucleotides. Right down here. This is our subunit for nucleic acids, which we'll talk about. But this is really just an overview of this chapter. So I don't want you to get too bogged down in this diagram. It's really more of just a roadmap. And I think if you look at this one at the end of the chapter, it'll make a lot more sense. So what you really need to understand throughout the process of this lecture is the idea of chaining together what we call monomers. Okay, so monomers are just any single thing that you can copy a bunch of times in order to make a polymer. So mono means one, poly means many. So you take one, you string a bunch of them together, and now you have many. So if you notice, we have this same monomer right here below, but we have it again and again. So there's 4 copies of this monomer inside of this single polymer. Okay? So your body and any organism really does this quintessentially. It does this all the time. It builds stuff up into polymers, and it also breaks stuff down back into its monomers. And so I just want you to understand that these are like Legos. You're really just putting Legos together and building something bigger. And you can take it apart later in order to build up other things. Now, when you take in food, you take in a whole bunch of stuff. Okay, and that's great. But sometimes what you take in isn't exactly what you want. So what your body does is it breaks those big things down into smaller things. Okay, so that's what we have here. This process this happens through catabolic reactions or catabolic pathways. Okay, so we're going to break down these big things, and we're going to get some energy out of that. We're going to lose some energy to heat. Anytime you change energy forms, it's a little inefficient. And then way down here, well, we have some energy that we have accounted for, but we haven't accounted for all that matter. So here we have a bunch of basic building blocks. These are our little Legos. Okay, so I had some, I ate a Lego house, I ate a Lego building and a Lego boat, and then I ate them and I broke them down, and now I just have the regular Lego bricks. Okay. Now, when I have all those bricks, I don't want those to go to waste, so I'm going to build those up into whatever it is that I need. Okay, so maybe instead of a building, I needed specifically a skyscraper. So I broke down the building and now I built a nice skyscraper. Or I broke down the house and I built a mansion. Okay, and anytime you're building anything, just like when you're building stuff with Legos, it takes energy. Right? And so some of that energy that we got from breaking stuff down, because remember, you're breaking bonds, and breaking bonds releases energy. So some of that energy is going to allow us to build stuff up, build up whatever we need. And now notice that these are not identical to what I took in. So I broke them down, changed them some, and built them back up. Okay? And when we build things, those are called anabolic pathways or anabolic reactions. So breaking down is catabolic, and building up is anabolic. Catabolic releases energy, anabolic requires energy. And I really think that the best way to think about this is in terms of building something like Legos, where if you want to build something cool, you really have to put in some energy, right? Now, this whole reaction web, if you will, where you're breaking stuff down, building stuff up, we call that metabolism. Of course, you've heard metabolism before. Now, again, anabolic reactions are when you take smaller things and you make larger things. But remember, that always takes energy, so we have to add some of that in there. And so in an example that you will see over and over and over again, we have condensation reactions. So here I have two monomers. Notice that they are identical. And I want to combine them to make one polymer. Well, really, it'll become a polymer later, but I want to combine them. Okay? In order to do that, I need to get rid of this hydrogen and this hydroxide right here. They're kind of in the way. Okay, so I need to get those out of there. So what I'm going to do as a cell is I'm just going to chop off, oops, sorry, not used to max. I'm going to chop off the hydroxide and the hydrogen ion. And those guys are going to go do their own thing, and they are actually going to combine into water, so H2O. Okay, and now I can connect this spot right here to this spot. And so now I've just added some train tracks, right? I like to think of it as train tracks. You're just building it and building it. And so every time that you're building stuff, you're going to take out some water. And so that's why we call it condensation reactions. So remember that when water on the outside of your glass condenses and forms droplets, you get water on the outside. So that's why we call them condensation reactions. So we do have a new covalent bond formed right here between these two parts of the monomer. Okay? And we can do this again and again and make a really, really long chain, or we can stop at 2, 3, whatever. Okay? Now, this is the same thing, just showed in a different kind of diagram. This has more intense molecules. Okay, but it's really the same exact idea. You're going to take out the parts of water and connect them into one long chain, and hey, look, I have water left over. Okay. Now, catabolic reactions, remember, that's the other side of the coin. We are breaking stuff down. Now, in order to break stuff down, we actually have to do the exact opposite. Instead of taking out water, now we have to add water in. Okay, so we call this hydrolysis, and that might sound silly, but hydro, of course, means water, and lysis is actually going to refer to cutting something. Okay, so if we're going to break it down, we're going to cut these bonds. We need to do that by inserting some water. Okay? And this won't happen by itself generally, so you need an enzyme that'll actually do this, and we're going to get more into that later. But same with anabolic reactions. Usually these things aren't going to happen on their own. You have to make them happen. Okay? But in this case, we actually release some energy, and then of course we get our little building blocks. So here's another example, but with more intense actual chemistry, but it's still fairly basic. It's the same exact railroad idea. Just take a look at this, pause the video, and just give it a chance. Try to compare this diagram to this previous one over here, okay? Because it's really the same thing. Now, this is just a nice little summary table. So in condensation reactions, you have to consume energy, you form covalent bonds, you take water out, and you take your monomers and chain them into polymers. Hydrolysis is the exact opposite in every single way. Energy gets released, covalent bonds get broken, water gets added, and polymers break down into monomers. Every single thing is the opposite. Now this takes us to our first major macromolecule, the nucleic acids, and here we have an image of DNA. So DNA and RNA are our two nucleic acids that we care about for this course. So DNA, of course, stands for deoxyribonucleic acid, which I know is a mouthful, and ribonucleic acid refers to RNA. Okay? You don't really need to learn to spell this, just be able to recognize it and use it once in a while just to practice. Okay? Now, These are polymers, which means they're long chains of monomers. But in our case, for the nucleic acids, those monomers are referred to as nucleotides. Okay, so I want you to keep that term in mind, nucleotides. And we link those nucleotides together with a phosphodiester bond, which sounds like a lot, but it's really, really simple when you get down to it. And we're going to do an activity in class where you actually build these yourself. So you're really going to get a lot of practice. But these nucleotides can be represented in this diagram. So this whole thing, I'm going to take a quick second, this whole thing would be our polymer, okay? And we know that polymers are made up of monomers. And in this case, our polymer is, well, I know that this is going to be DNA, but you could also just put nucleic acid, because RNA looks very similar. Now, here's our polymer, and it's made up of monomers. So here I'm going to put monomer. I'm just going to put mono. Okay? And this is a nucleotide. Nucleotide. Now, not all nucleotides are the same. So we have a nucleotide labeled C, a nucleotide labeled G, A, and T. And you'll notice that there's different shapes going on here. We'll get to that. Okay? But every single nucleotide, so every single C, G, T, and A, can be broken down into 3 basic components. Okay? You have a phosphate group. That's going to look the same across all nucleotides. You have a sugar. That's going to look the same across all nucleotides. And then you have what we call a nitrogenous base. Now, a nitrogenous base, this is where you actually decide what nucleotide it's going to be. This is where you decide if it's going to be A, T, C, or G. So the one diagrammed here is adenine. That's our A. So that's why it's labeled as such down here. But this could just have easily been thymine, cytosine, or guanine. Okay? Now, you'll notice that these sugars have 5 rings and all of that stuff, and that's great, and your book goes really in depth on it. But for now, I just want to focus on the overall structure and how they kind of link together. I really like this form of simplified diagram. Okay, so we have a phosphate group here, we have a sugar here, very clearly a sugar because it's a pentagon, and here we have our nice little tag or our nice little label. This is our nitrogenous base, so I could just put T in here if I was doing this by hand. All right. Now, DNA is going to have A, T, C, or G, adenine, thymine, cytosine, or guanine. RNA, however, does not have thymine. Instead, it has uracil. Okay, so uracil is unique to RNA, and so if I ever showed you a nucleic acid that had a bunch of letters, you should be able to tell me whether it is RNA or DNA. Because if it's RNA, that means it probably has a U in there somewhere, and if it's DNA, it probably has a T in there somewhere. Okay? Now, RNA looks like this. Well, it can look like a lot of things, but here's a basic. Okay? You'll notice that it looks a lot like half of the DNA, and that's actually basically what it is. Okay, so DNA is in this what we call the double helix structure. Surely you've heard that before. Okay? And RNA is single-stranded. So this is made, the DNA is made up of 2 strands, whereas the RNA is made up of only a single strand. Okay, so I would need like another RNA to put on here, but RNA just doesn't do that. Okay, it doesn't play well with itself like that. So there are a couple other differences. Those are highlighted in this chart here. So as I said, DNA is double-stranded, RNA is single-stranded. DNA uses a deoxyribose sugar, that's why it's D instead of R, whereas RNA uses ribose sugar. And then, of course, the T instead of the U for DNA and the U instead of the T for DNA. And then here's just what those differences look like on an actual nucleotide molecule diagram. Okay. So next I want to move on to proteins. We're just kind of doing like a survey of each macromolecule for now. So proteins are really, really awesome in your body. Okay, or in any organism, I should say. So proteins are made up of amino acids. Okay, so amino acids would be considered the monomer of proteins. Okay, amino acids will link together and they form a polypeptide, also known as a protein. And we link those things together via a peptide bond. So if I have amino acid here, and I have an amino acid here, I'll draw a different shape because maybe it's a different amino acid. Well, they're linked together via a peptide bond. Okay? That's exactly what this diagram here is showing you, but I'm trying to take out all of the chemistry for now so that you can just understand what's kind of the concept and then look at the chemistry. Okay? So every single amino acid contains some of the same stuff, just like nucleotides all had that sugar and they all had that phosphate group. Now every single amino acid will have an amino group. Okay, that's right here. It's going to have a carboxyl group, which is way over here. It's going to have an H group. Sometimes you won't see that referred to. Okay, it really depends on the textbook or the article that you're reading, anything like that. But you'll always have some kind of an R group. Okay, so we call it an R group, and that's just kind of a catch-all because there's a whole bunch of different R's, if you will. This can be a lot of different things. Okay, but if I were to just kind of boil it down, here's an amino acid. This is the schematic for an amino acid. I have an amine group, a carboxyl group, and an R group. And then, of course, there's a carbon and a hydrogen here. Okay? The R group has 20 different possibilities. Okay? So there are 20 unique amino acids. This is the same, and this is the same for all of them, but this part is different. Okay? Now, these R groups really determine what these amino acids are going to be like. Okay, so if you look throughout here, I don't need you to memorize any of this, not even the names, not even the little symbols. You'll have all of that access to you whenever you need to use it, including on the AP test. But just know that the R group, really determines what it is because it's the only unique thing about it. Like the amino acids are all the same except for the R group. So the R group can make an amino acid hydrophilic. That means that it can dissolve in water. Hydrophobic, which means it can't dissolve in water or other polar things. And ionic, which means that it has a charge on it. Okay, so we see some ionic over here. And ultimately, we call all of these things side chains. So all those R groups are side chains. I just want you to kind of get used to that terminology as well, because your textbook uses it. Now, these polypeptides get put together by the ribosome. So here's our ribosome. It's this big, weird molecule. And we're going to come back to this a lot when we talk about gene expression. So you're going to learn this whole process. But for now, I have different amino acids. They have abbreviations right here. And they're just getting linked together. And so just think to yourself, what would you call this bond? And this bond, and this bond. Well, what can we call that? Well, these are forming a polypeptide. These are peptide bonds. Okay, so peptide always refers to something dealing with a protein. Okay? You can think of it because P and P, they both start with P. So proteins can be really, really complex, but we need to look at them at 4 different levels because they're so complex. We have the primary level, the secondary level, the tertiary level, sometimes the quaternary level. Okay, so this is like 1, and you'll see this terminology a lot. 1 with a little degree symbol, 2 with a degree symbol. This is just saying the same thing, except it's easier to write. All right. Now, these things all build on each other. So your primary structure determines your secondary structure, which determines your tertiary, etc. But we need to talk about each of them individually. So your primary structure is literally just what amino acids are there and in what order. So you could, if you were trying to say the primary structure, you could say, oh, it's this amino acid connected to this amino acid connected to, right? So it's just what order? Maybe this one repeats, doesn't matter. Okay? And so this would be our primary structure. Okay? The secondary structure is where you start to actually see things interacting. Okay? So we start to see hydrogen bonds. Remember those weak bonds from when we talked about water? We see these hydrogen bonds forming between our amino acids. Okay? Now that causes our proteins to take on different shapes. They're not just a flat line anymore. Okay, so you might have weak hydrogen bonds going on here, and that causes the thing to fold up in a weird way. Now it looks like a folding screen. Okay. Now there's 2 different examples that your textbook talks about. There's alpha helices, which has hydrogen bonds between every 4th amino acid. Okay, hydrogen bonds between every 4th amino acid. And then this causes a nice helix structure. And there's beta sheets, which has adjacent regions forming hydrogen bonds and laying them side by side. And I don't want you to get bogged down in the differences between those. Those just deter, like if you needed a protein to perform a specific function, you would need, maybe you need it to be a nice structure, right? You need it to be very structural, very strong. Okay, then you might have some alpha helices. And that's great, but you really don't need to memorize it. And it's a little bit too much for the course, okay, for our purposes of this course. Tertiary structure, so this is our third level. Okay. This will describe how your polypeptides fold. Okay, so if you look here, I started with my primary, and then I started to form some sheets and some helices in my secondary, and then those start to interact to form my tertiary structure. Okay, so each time it's really just building on itself. So I took my primary, I started to add some hydrogen bonds. And then I took my hydrogen bonds that formed sheets and helices, and now I'm going to connect those together in some given way in order to form my tertiary structure. Okay. Now, these are a little more complicated, so there can be hydrogen bonds. Sometimes this becomes this just through hydrogen bonds. Sometimes it's ionic bonds, so there's a positive and a negative somewhere. Maybe there's a positive here and a negative up here, and they're going to interact and fold around in this weird thing. Okay. Or there can be disulfide bridges, so di means two, sulfide refers to sulfur. Okay. And that's just basically where you actually have a covalent bond forming between two different areas. So maybe like here and here. Okay. And so that's going to affect the 3D shape. You do not need to memorize any combinations or any specific protein structures. Just be familiar with these three ways by which they may occur. Okay. Now, your tertiary structure is really where you can start to see it functioning. Okay, some proteins stop at the tertiary level. Some don't even have a quaternary level. Okay, but these are three-dimensional things, and that's really hard to picture sometimes. So you'll often see them represented as like these giant amorphous lumps. Looks like a weird mutated piece of popcorn. Okay, but there are specific regions on here that really matter to us. Okay, and some regions on a protein will allow stuff to bind there. And some regions of a protein might let stuff pass through. And we're going to get to that. But the final structures, this is our quaternary structure. And this doesn't always occur. But this is if you take multiple polypeptides. So you take multiple chains of amino acids and you have them interact in some way. Okay? And there's not a ton of examples that you need to be familiar with. The one that you'll typically see is hemoglobin. So here we have a hemoglobin molecule, and it's made up of four different polypeptide chains interacting. Okay, so you have a beta chain 1, a beta chain 2, an alpha chain 1, and an alpha chain 2. They're all just interacting in different ways. You can see those interactions here in this diagram. This is like the chemical form of it, but don't worry about that. And oxygen is crucial to hemoglobin's function, and so if oxygen is absent, these don't hang out together anymore, okay, because of a consequence of how they're connected. Okay. All right, so normally the 3D shape, remember these are 3D, of a protein gets stabilized by your bonds, right? That's what I was talking about when it was folding and doing whatever it was doing. But sometimes extreme conditions that the protein's not ready for, might cause those bonds to break. So maybe it got too hot and your hydrogen bonds broke. Okay? If that happens, well, this breaks, and now this piece is just kind of going to fly up and do its own thing, and they're not connected anymore. And that means that this protein can't do whatever it's supposed to do. Okay? Maybe this ionic bond gets broken. Okay? Well, maybe a protein got in water when it shouldn't get in water, and now the ionic bond has been disrupted because we know that water likes to break up ions. Okay? Now, disulfide bridges can break too, but they're pretty strong because that's a covalent bond, right? That's the strongest of those three. Whenever a protein loses its structure, we call that denaturation. Denaturation, depending on who you ask. It's a denatured protein, okay? And no longer has the correct shape. This can happen because the pH got messed up. It can happen because temperatures got messed up. A whole bunch of things. Maybe it lost a piece, right? Or had a mutation that caused it to be messed up. Okay. And these things, depending on the protein, can easily go back when the conditions get ready again, or sometimes it's harder. And we'll talk about that later. But when you cook eggs, okay, so eggs, we know, when we think of protein, one of the foods that you probably think of is eggs, right? When you're adding heat energy, you're actually causing all of the proteins in the egg white and the yolk to denature, okay? This is why the egg actually becomes more solid. It becomes insoluble, which means it cannot be dissolved anymore, and it starts to stand out, okay? Now, our third macromolecule is lipids. Lipids are also known as fats, and these are going to be quintessential when we start talking about cells. Okay? And this is a triglycerol molecule that we're seeing here. It consists of a glycerol and 3 fatty acid chains, and we're going to get back to that in a second. But just start to be familiar. I want you to take a look at this structure. Notice what atoms you're seeing, okay, and what the overall structure might look like. There is some diversity. Okay. But this is a really good diagram to show you what a fatty acid chain really will often look like, or lipid will look like. Okay. So here, you'll notice that there are a lot of carbon and hydrogen, and they like to hang out. Okay. So I have carbon and hydrogen bonds all over, and carbon, of course, because it's a long chain, but carbon and hydrogen bonds all over the place. Now, if you remember from chemistry, carbon-hydrogen bonds are non-polar, right? The electrons like to hang out equally with hydrogen as they do with carbon. So this is not going to be polar. Okay? There might be some polarity going on over here because oxygen is very electronegative, but overall, lipids are very non-polar because of this whole section. Look how huge that is. So this is why lipids typically won't dissolve in water without a little help. Okay, so if you put butter in water, you'll typically, if it's melted butter, you'll see one float on top of the other. Okay. Now, this slide is really trying to show us the difference between saturated fats and unsaturated fats, and that's important, but I don't want to delve on it too much. Basically, saturated fats are full. That's what saturated means. It's full. So if you're saturated with water, you're full of water, right? So these guys are full of hydrogen. So every single opportunity for a hydrogen, they've got one. However, unsaturated fats have some places, like what's going on right here and right here, that don't have hydrogens where it, could have. So it could look a lot like this upper one, right? This higher one. But instead, these carbons decided to have a double bond. So anytime you have a double bond going on, or even a triple bond, depending, that means that you're not going to have hydrogens when you could have had hydrogens, because each carbon atom can only have 4 bonds. Okay, so we call that unsaturated. And that's going to affect what your lipid actually looks like. So saturated fatty acids, you've heard of saturated fats. They're going to be really, really thick, right? So like butter is very high in saturated fat. Whereas unsaturated fats are going to be more fluid, like olive oil, for instance. Okay? I don't want you to memorize any specific saturated or unsaturated fats. But as we talked about, lipids generally are nonpolar because of all those carbon-hydrogen bonds. But phospholipids are a little different. They have a polar area and they have a nonpolar area. Okay? This is really the crux of why cells work right here. And we're going to come back to it, of course, because we have to talk about cells in great depth. But they have a head, so that's this guy right here, right? Let's put a little face on it, and that's our head. And then it has the tail region. So there's a head region and a tail region. Okay? The head region is hydrophilic, so that means it is polar. And the tail region is hydrophobic. So you'll notice that all the hydrophobic parts like to hang out with each other. Okay? So all these tails are near each other. That's because like dissolves like. Similarly, all the hydrophilic heads like to hang out with each other, and they don't like to get in here. Okay? That's just a consequence of the chemistry of them. That's good for a primer of lipids. And so I want to move on to carbs because we really are just kind of touching it, and we'll get back to lipids when we talk about lipid bilayers, when we talk about cells. Okay. So carbs are our final macromolecule that we're going to talk about. Carbs are also referred to as polysaccharides, and they're made up of sugar monomers or monosaccharides. So we have the same poly and mono thing going on here as we had with polymers and monomers, and polysaccharide is a polymer and monosaccharide is a monomer. Okay? And those are joined by condensation reactions, just like every single one of the other macromolecules. Okay? And their bonds and their structure are also going to determine their properties. So you should be noticing some trends depending on what they are, shaped like because of their chemistry, or what atoms are involved and where. That's going to determine what they do. Okay? So monosaccharides are just single sugars, right? And the classic example that we're going to talk a lot about is glucose. So here's a glucose molecule here. However, you can put these guys together. So if you start to put together multiple monosaccharides through the same process that we do everything else with when we combine stuff, condensation, remember this would be an anabolic pathway or reaction, they're going to form a disaccharide. So hey, look, I now have two sugars hanging out with each other. If only I had a way to say two sugars in really fancy science language. Oh, di means two, and saccharide, actually means sugar. So a monosaccharide is a single sugar, right? That's why we have it right here. And a disaccharide is two sugars put together. And then you can do even more and make a polysaccharide. Remember, poly means many, so it usually means three or more. Okay? And so here are some examples down here of different monosaccharides, disaccharides, and polysaccharides. Again, don't memorize it. You should know that glucose is a monosaccharide, but the rest will come as we start to talk about the structure and function of these different things. Okay, so sucrose and maltose, I just kind of want to get you to look at some of these chains. So when you start to see these things chain, you start to see, oh, look, we have hexagon next to a hexagon, or it might be a pentagon, right? You start to see these same shapes over and over and over again. Okay? And here's an even more simplified example. So notice that the same shape is being repeated like crazy. That's how you know it's a polymer. And specifically, the shape here is a hexagon. Hexagons almost always are going to indicate that it's a sugar. Okay? So that's a really, really good tell. If I took away this glycogen label, or this starch label, or this cellulose label, I would still, by the end of this, expect you to be able to tell me, oh, this is definitely a carbohydrate of some kind, because look at all these sugars. Okay? You don't need to memorize what they all look like. Just recognize, oh, look, that probably is a sugar because I see a lot of hexagons. Okay? I'm going to skip over cellulose for now because we are going to talk about that a lot more later, excuse me. And then same with starch. Okay. And this is our last little topic I want to cover in this lecture. This is directionality, and I'm just going to barely touch on it. Okay, directionality is, of course, having a direction, and this is really going to matter for some of these biomolecules. Specifically, I need you to start thinking about it in terms of nucleic acids. So nucleic acids, including DNA, which is inarguably the most important molecule that you're going to learn about in biology, okay, is going to have different ends. It has a 5' end, okay, 5' end, and a 3' end. Now, those are called that because of the carbons involved in the sugar. So if you'll notice, we have a naming system, a naming convention for each of these carbons. So if you remember from chemistry, every one of these little vertices is going to be a carbon. Okay, here's the first carbon. Here's the second carbon. Here's the third. Here's the fourth. Here's the fifth. So the prime just says, hey, it's a carbon. And the number just tells you which one. And I don't need you to memorize like, oh, we go clockwise from the base. I don't need you to memorize that. I just need you to know that there is a 5' carbon and a 3' carbon. And really, I like to think of it as an arrow. Okay? So we're going to be talking about directionality in the context of DNA a lot, because things like to go from 5' to 3'. So I just, for now, just need you to realize that these DNA and RNA molecules have directionality. Okay? They have a specific direction. So here I have a DNA molecule that's being separated into its two strands. The blue strands are the two strands that were combined before and are combined further downstream. Okay? And then the red strand is the DNA that's getting made. And we'll get back to that, but it can only go in one direction. And so that's going to become really important. Okay? Proteins also have some directionality. Okay? So remember that proteins have your amino end and your carboxyl end on the amino acids themselves. Okay? And then that, as a consequence, affects the protein. So the protein itself also has an amino end and a carboxyl end. Don't need you to memorize which, but you should be able to recognize the N-terminus side of a protein and the C-terminus side of the protein. It's really, really obvious. If I showed you a huge polypeptide or a protein right here, you should be able to say which one is the N-terminus because it always looks like this. It has this NH3 plus at the end. Okay? And this NH3 plus right here. Look, N-terminus because it has a nitrogen. The C-terminus side is always going to be on the other side of that. Okay? And it has a carbon. That's why it's the C-terminus. Okay? So just be able to recognize those. And then here we have our R groups, our side chains from our amino acids. Okay? And this is also really important for carbs. So carbs can kind of their structure is determined by their function is determined by their structure, just like anything else. But sometimes you won't really need a strong carbohydrate maybe to use for structural support. And so you would really want to form it in one way rather than another. And so again, I don't want you to memorize any of that. I just want you to be familiar with directionality as it applies to carbohydrates and proteins and, of course, nucleic acids. Okay? And then if you need a little bit of help understanding any of that, here are some useful links. Okay, so I am a huge fan of Crash Course, but Crash Course has a nice episode on biological molecules, which is what we talked about. Bozeman Science is great and has some proteins that they talk about in the protein video. And then Ricochet Science has one video for each of the major macromolecules with the exception of proteins, because I put the Bozeman on here because I actually really like the Bozeman one. Okay, so if you have any questions, let me know, but that is going to be the end for macromolecules.